bubble back over the porch.
Yep.
Alright, the ship's starting
Yeah,
to
start
steady.
moving again.
Roger.
Sorry about that, guys.
Bridge
Yeah.
now.
Failure.
No problem.
Request 20-meter step, bearing 320.
One of those little sea cucumbers floating by.
Tiny one. So small.
Alright. All those viewing, can you see high pack again?
Yes.
Awesome.
And a note about the crinoids that we've been discussing. We've talked about the stalked crinoids versus the unstocked crinoids. There are these crinoids that in their early stages of life have a stalk that they shed. But the crinoids that we're seeing on the stalks that are adult forms, they're not going to be coming off of their stem and fluttering away the same way that the unstocked crinoids do. And the unstocked crinoids is what we refer to as our feather stars. And you may have been hearing stalked crinoids referred to as sea lilies before, but we call them crinoids around these parts.
Bridge nav. Request track a line, bearing 320 at 0.3 knots.
There's a lot of these little
What's up?
cucumbers around this area. Jamie.
Oh.
Hi.
Hi.
For the sea cucumber that lives in the water column versus being on the seafloor, are they just pulling the marine snow out of the water versus picking up the sediment directly from the seafloor like their relatives?
I believe so, yes. So, their benthic relatives have little tube feet.
Yeah.
And they have a ring of them around their mouth. And they stick them down on top of the sediment, pick up a little handful, and then pop it in their mouth, and they do that alternatingly with all their tube feet. I think the pelagic ones, though, somehow just bring the water into their mouths and filter from that. I'm not 100% sure, though, so if someone knows otherwise, let us know.
There's another sponge, I believe. Right to the top right.
What's that?
Well spotted.
Top right of the screen.
Sponge. Okay.
Bridge nav.
Stop them there, yeah.
Request we hold position.
Full wide on the Zeus. Thanks.
There's a little something living in it.
This is a better sponge anyways.
I like the other one.
Yeah, well.
The one that got away.
Yeah. Think about that all night.
Yep. That was a nature paper in one sponge.
This is our redemption sponge.
You want to image it first?
Yes, please.
Okay, zoom in video.
Now, is that something growing off it?
Looks like a little anemone.
Mm-hmm.
Told you this one was cooler.
Bonus.
2-for-1 deal.
Okay, come wide.
Full wide.
You thinking about grabbing this one since it's a little
Yeah,
bit bigger?
might
Yeah.
be a bit of a,
Yeah.
might get a bit smashed up, but.
Would it be alright to come a little closer with Argus Zeus?
You can zoom in there. Sure. Where's this guy gonna go?
Somewhere where it's less
Is
likely
it-
to float away.
Yeah,
maybe.
it's probably going to be pretty floaty, so maybe the small starboard compartment, or if you think that's too risky, let's put it in the forward bio box.
Okay. There is a way that you can get this to stop. Hold on. It's closing. If you get this perfectly still. Nope. It's hard. Zoom in there a little bit, video. Thanks.
Nice.
Skillfully done.
That was the next level sampling.
I don't have them yet.
Okay.
All right. Should stay there. Or not. No, that's opening.
Let's put it in the forward bio box if you think
Yeah, okay.
it looks
Forward
pretty
box out.
floaty.
Do a tray out.
Yeah. Can you rack the camera in?
Yeah.
Which compartment is
Your
free?
Zeus?
Either.
Okay. Yeah,
Whichever's
Zeus, rack it
easier.
back.
On here.
On the, watch. You can do that on the GUI as well.
I gotcha.
Coming wide on Argus Zeus.
Do a tray in.
Nice one, Mike.
I don't know if I call it nice. It's done anyway. Yeah, the just controls here for the camera and stuff like that.
Okay.
So
Which
you,
one's? Oh,
so you can do all the rack
oh,
and
PNT.
pan and tilt up and
Gotcha.
down and all that whole stuff.
PNT retract. Okay.
New,
Yeah, watch this rocker because it could be something
Now,
else could
can
be
you drop a
selected there.
target
Really?
here?
Yeah, like this could be a bunch of different functions.
yes,
Ah.
That
what
is
sample number is
sample
Okay.
036.
Thank you. Sorry.
that? 036, roger that.
The more you know.
I'm gonna reset DVL real quick here.
Sure. Okay, we can move the ship when you're ready.
Roger that. Alright, all reset and back on the move. Bridge nav. Bridge nav.
I'm seeing some of the string-like
Request
tracks.
we track a line bearing 320 at 0.3 knots. Thank you.
So we just collected a sponge sample with an associated sea anemone on it as we move up Dragon's Back Seamount here off of Palmyra Atoll. Are we approaching the first peak of this multi-peaked seamount?
Yeah, that's where we're heading, but we're a ways away from it. Chris can tell us the range.
Yep. So from the peak that we're shooting for, we are currently a little under 450 meters away. And still a little bit of upslope to go.
Is that a critter? Maybe that's just a rock. Never mind.
I know my eyes are doing the same thing.
Anything that's slightly darker, I assume is a critter.
Is that another little sponge sticking up off of the rock there? Above the lasers? Maybe not.
Where are you looking? To the left?
Where is
To the left of the lasers.
Oh, yeah.
Little right.
Yeah.
I've been seeing these platy white things hanging
I don't
off
see
the
it.
rocks.
It's below the lasers now, on that like curvy sort of, the end of that rock curve.
7 o'clock.
7 o'clock.
Going further from it now, on the other side of that curve.
Oh,
It's hard to view
Yeah.
from this angle. It was
it
easier
here
to see.
somewhere?
Yeah, it was right about there. Can't really see it anymore. I don't know what it was.
There is a critter to about 10 o'clock from the lasers. It's like a stalked, maybe clinoid.
Oh, I see what you're talking about, Kevin. In the back.
Oh, yeah, that might be a stalked crinoid. So if you go straight left from the lasers right now toward the edge of the screen, there's something yellow. Is that what you're talking
Oh,
about? Yeah.
zoom in there, video.
Full zoom.
Yeah, that looks like a stalked crinoid.
All right, come wide.
sea cucumber right there by the lasers.
Can we get a zoom on that fish?
Actually, yeah. I think
Roger.
Is that a shrimp or?
Okay, go ahead, video.
Pretty big shrimp if it is one.
Oh yeah.
Yeah.
Wow, it is, huh?
That's interesting. Most of the shrimp that are on the larger side we've been seeing are all red, and this one has that big white patch
yeah.
on the back.
And it seems to be using its walking legs.
Yeah, instead of its swimming legs.
Thank you.
And if you're watching on nautiluslive.org, remember that you can send in your science questions
For what?
into that feed
Roger.
below the video. We'll do our best to answer them here in the van. So again, we're moving up this seamount formation that we've been affectionately calling Dragon's Back Seamount, south of Palmyra Atoll.
Is Dragon's Back something that was created, the name created in the last few days, or is that something?
I heard it from Steve yesterday.
Cool.
Yeah, it was when Steve was looking at the map, he was trying to think of names, and he made that one. And I said, well, if you say it enough, it'll stick.
And so he came
And
up and he
so-
said it to everyone.
That's how it works, right?
Yeah.
I told
so
him
you
if
can actually propose seamount names. Like in a manuscript I've written, I added basically like 25 seamount names.
Oh,
for the
look at
seamounts
that.
I want addressed, but it will go to a committee somewhere.
Yeah.
In like somewhere in Europe, I don't remember. And then they may keep it, or they may just change it and name it after some old European scientist.
And that's why I'm saying it's affectionately called Dragon's Back Seamount here.
Not officially.
Yeah.
I'm gonna reset DVL. It's getting away from us.
Sure.
How do they keep track of which ones need to be brought up to committee to be approved or dismissed?
Yeah, that's a good question. I don't know. So no one has contacted me. So I'm assuming all my names stand, and they haven't discovered it.
Can I ask you to list off a few names, perhaps some of your favorites that you've assigned?
Salaga. I named them all after. So we're in the Tuvalu region. So I went and looked up Tuvaluan mythology, and I
Cool.
named them all after major characters in those mythologies, because obviously I want to name it after the locals. So I'm not gonna call it like Oregon Seamount.
Conrad
Good
Mount.
call.
Conrad. Yeah, definitely not gonna name it after myself, but it would be pretty cool to have a seamount named after you.
Yeah.
Someday. You
Well-
will.
Someday.
Steve didn't like my suggestion of the Steve Seamount, so
Steve Mount.
I mean he might have liked it, but I guess he thought that Dragon's Back was better. Whatever.
Sounds kind of cooler.
Yeah.
I don't know. I can relate to Steve though.
Steve Seamount has a ring to it, doesn't
I like
it?
it.
Yeah.
Yeah, I like it.
And it looks like we've got a comment written in from the Museum of Comparative Zoology Invertebrate Department asking to keep an eye out for small crustaceans in the samples because they're looking forward to receiving amphipods.
Cool.
That'd be cool if we could sample one off of those. Have we seen some on fish or something? Those little amphipods?
Amphipods, yeah.
Yeah.
I think some of them
The
are
ones on
the
fish, though, are
parasitic.
usually isopods.
Oh,
Oh,
isopod. Oh,
isopods.
my bad.
Could we zoom on that purplish stalked, maybe stalked structure to the left of the lasers, about midway between lasers and the edge of the screen?
Okay, I don't see it yet.
Okay, it's
Right
just
above
above
it.
the
Oh, I got it.
lasers now.
Yep. Yeah, go ahead, video.
Coming in. Cucumber.
Maybe a sea cucumber, actually. Definitely not stalked, though. That was an optical illusion.
But you never know down here
Full
on the
zoom.
deep sea.
More like stocky.
Stocky.
Poor guy.
So is a squat lobster called a squat lobster because it squats or because it is squat?
I don't understand the question. Just like before when you were pronouncing squat lobster differently, I was really struggling to...
So squat can be a verb or an adjective, right?
Okay.
So-
In this case, it would be the adjective form.
Okay.
Because I think it's called a squat lobster because it looks somewhat like a lobster if its tail is out straight, but it's not a lobster, just to reiterate that. It is a crab, but it does have some lobster-like visual qualities. So it's got, if you imagined a lobster and it tucked its tail underneath its body,
What's that?
that's basically the form the squat lobster holds.
The DVL?
So, yeah, it's sort of got the tail end tucked underneath, which gives it more of a squatty appearance than a full-size lobster, but it is a crab.
It might be the slope. I don't know.
Cool.
I'm
I was
seeing
very
4
curious.
beams fairly constantly, so it should be fine, but... Yeah.
Oh, is that
Another
a
floating cucumber.
so cool.
Potentially some critter directly above the lasers on in between 2 rocks, or is that sediment? I can't see it.
Is it just me, or does it seem like there's bigger pieces of rock that we're starting to come across here?
Yeah, as we get towards the summit, I think we're seeing more of like an intact, thicker flows.
Again, thank you to all the viewers joining us on nautiluslive.org. Remember that you can send in your science questions below the video feed in that white bar. Questions about what we're seeing, where we're going, life at sea. Send them in. We'll do our best to answer them for you.
I'm going to start a gauge check and be mindful of everything
Sure,
going
yeah.
on.
Got a question here about the coolest part of our jobs. I mean, speaking as an educator here on the ship, the coolest part of my job is being able to be out here and experiencing this firsthand, getting to know scientists and engineers that work on this stuff, and really exploring the seafloor like this is something that, you know, I've always been interested in just from a personal perspective, but especially as an educator, being able to take this experience back to my audiences and my groups that I work with is really exciting. I was a fellow last year. This is my second year with Nautilus, and so over the last year, it's been awesome to be able to bring that firsthand experience to the floor of my museum and talking with audiences from all over the place about this experience and connecting engineering and all the different aspects of science together in what is a really interdisciplinary endeavor that happens here.
I have a question for you about that.
Yeah.
So did you work at the museum before you came out on Nautilus?
I did, yeah. I've been working at the museum. I just hit my 5-year anniversary
Okay.
at the Franklin Institute. Yeah.
So my question is, can you say anything about different reactions or connections that you can get with people when you're talking about your experience out here versus before you came out and you weren't sharing that type of information with them?
Yeah. So just for a little more context, the museum that I work at is the Franklin Institute, and it is mostly a technology-based museum, a lot of what we talk about, but also human anatomy. So those are the two main topics that we do on the floor of the museum and in our workshops and all of that. And I was trying to tie in marine science wherever I could because that was of personal interest to me. But after this experience and really having access to all the educational materials that are developed by Nautilus Live, which, by the way, anybody at home watching, you can also access these materials, from full lesson plans to hands-on activities that have been developed by the education team and by science communication fellows from all over the country. Once I really had that context, I've been able to build more in-depth experiences around it. For example, I last summer developed a training program for our high school volunteers, and in the past we've trained them on everything in the museum and all of that. But I was able to add in these live interactions with the ship during our training week. And it really made a big difference in getting them engaged around what science communication and science education is as a field, instead of just, this summer volunteering thing that they were doing. So that is one way that it's really deepened the experience. But also we do something called Random Acts of Science on the museum floor, where the team of educators that I'm on, there's 4 of us, along with some of our colleagues. We go down onto the museum floor and have an hour and a half to really just present whatever we want at our, at one of our stations. And I have been weaving in more and more of my experience working directly with scientists who are collecting these samples, who are creating these dive plans, because before it was pretty abstract to me. It was something I watched, it was something I was interested in, but I never had that firsthand experience of watching people create a dive plan, watching people look at the sonar mapping and try and understand what the features might, show us biologically. And so I hope that makes sense. I feel like I'm going off on many different tangents now.
Yeah, no, that's very interesting. Thank you.
Yeah, but it's been awesome to weave it all together. And it looks like we've got some of my volunteers actually watching right now, which is awesome. Thank you guys for tuning in, and I'm excited to be talking to you with a live interaction next week. Got a question for you, Chris. What is our distance between Palmyra Atoll, the proper area above the water, and where we are now?
And where we are currently?
Yeah.
Give me just a second. Might take some zooming out.
Yeah, do what you need to do.
Might take a lot of zooming out.
If you're watching on the quad view, you can follow along with Chris's zooming.
So, let me see if it's in here. Alright. So, Palmyra's right at the top of the screen now. And from us, let's put it in nautical miles. Let's see. Did I select it? Yeah, it's selected. If this is right, 42.5 nautical miles.
Got it. Thank you.
It should make sense because it was about 4.5 hours for us to get over here going 10 to 12 knots.
Mm-hmm.
Am I clear to use the bubble?
Yeah, go ahead.
Thank you.
So I'm starting to look for rock samples now. Just a heads up, I might call out a pile if I see one that I like.
Yep.
Does that mean we're peaking on the first peak here?
We're not there yet, but
Starting to
it
get
looks
there?
like it's pretty steep here. And I find on the steeper slopes, although it's harder to sample,
Mm-hmm.
you tend to get kind of more talus. There's something to the right, some critter on a rock. I think it's crinoid on a dark rock on the bottom.
Oh, I see it. It looks like a brittle star.
Oh, it's brittle stuff.
You want to have a look at that one? Might be a bit
I can't see. Oh, I see what you're talking about.
kind of going by it here now.
Oh.
Yeah, it's okay if it's not doable.
Go ahead and
I
reset
think I
DVL.
just
Sure.
saw something go off to the right of the screen too. Might have been possibly a coral. I'm not sure.
Yeah, I'm going to have to push ahead a little bit.
Yep, there's something right there in the middle.
Whereabouts?
To your left of the lasers.
I'll stop the ship there, Chris.
Yep. Bridge now.
Something in here?
That red. Oh, I'm talking about the life one. The red guy to the bottom right of the laser.
So he wasn't talking about a collection.
Oh, no, I was
thought
talking
we were
about
looking
rocks,
for
sorry.
rocks to pick up.
I got excited. I saw some life.
Never mind. We can move the ship, Chris. Sorry. Zoom in there.
Coming in.
Bridge now. Never mind.
Nice
We are all
anemone.
set. Thank you.
Okay, for what? Yeah, sorry, are we looking for, we sniffing around for rock samples now?
Oh, yeah, we are.
Okay.
If we see a nice kind of a pile of loose rocks.
Confusing us looking at the biology.
Sorry. I'll go back to ignoring the biology.
Only rocks.
There's some more biology to the left. I can't do it.
Just gonna have to try and get back out in front here.
Hey, Kevin.
That's me.
Why would we be more interested in taking the samples from towards the peaks versus other areas of these seamounts?
That's a good question. Typically, I don't like taking samples from towards the peaks. I like the base because they're kind of curious when the seamount started to form more than when it finished. But in this case, I want to make sure we're getting rocks directly from this kind of individual peak. And Rob put in his notes that he wanted them from the peak. So I'm not going to supersede that.
Got it.
We've got 4 peaks total, right?
Yeah.
So you can do base and peak
There's some nice.
of each.
Nice bits in here.
Yeah, that looks good if we can.
All right, stop ship there, Chris.
Roger that. Bridge now.
Full wide,
Request hold
please.
position. Thank you.
See one you like?
Yeah, can we get the one right to the right of the laser? That bigger
Yeah.
one?
Zoom in there.
Coming in.
Let me know when you're good for imaging.
We're good.
All right.
That
Couple
one might be
wider.
stuck.
Yeah, it might be. One of them has to be loose, though.
Show me the arm at the bowl camera. No, sir.
Alright, well, let's just find whichever one in that area is loose. If it,
This one?
hopefully, yeah.
I don't know.
Oh man, they might just be the parts of the pillow
Yeah,
that's
I
all
think
popping
so.
up.
It's all just the lobates.
Tricky rocks.
We've been bamboozled.
All right, I gotta go too.
Alright. Thank you for trying.
So you're thinking instead of being loose pieces, these are just sort of poking out through the sediment as from a larger piece?
Correct. Yeah.
Bridge now.
Life.
Yep, sea cucumber.
Request we continue tracking our line. Thank you.
I want to see some corals.
Some nice elongated pillows.
Yeah. Probably not the best for dating. Hopefully they have phenicus. I don't like things that are fine-grained and glassy.
Does that make what you do to date them harder to do?
Yeah, it makes it so they're not, typically not dateable.
Ah,
Yeah.
undateable rocks.
Are we still doing 0.3 with the ship?
Too much baggage.
We are currently coming back up to speed on our line though.
Yeah. Take them down to 0.2.
Roger that.
Two, yeah, there's a longer one.
Bridge now. Request we decrease speed to 0.2 knots. Thank you.
Now, if you've been watching this feed for a while, you might notice that we do have watch changes. We got groups of different people that come in every 4 hours and
Look at this
change
nice
over.
fish.
Ooh.
Zoom in there, video.
That's a hagfish. Oh wait, no, it's not a hagfish. It's
Is
an
that a
eel-like
Bathio
fish.
Euroconger?
I don't know. Is it an eel-like fish or a fish like eel? Wasn't that the debate last time? I think we decided if it is a Bathio Euroconger that it was a fish like eel.
Yeah.
It's giving us a nice side view here.
Fish like eel. I'm gonna come in a little tighter, yeah.
Yeah.
Nice.
Smile.
See ya. Wow. Nice flying. That's full zoom.
All right, that was awesome. Thank you very much.
Okay.
Coming wide.
Oh,
And
it's
so is
opened its
he.
mouth. Ah.
Okay, I'll come wide.
Whoa.
Yawn.
Is that yawn?
I feel you.
Do they yawn in the ocean?
Maybe it was threatening us.
DVL's not behaving at all, is it?
No, it is not. I can just switch to USBL if you
Sure,
want.
yeah. And try it at the top, see if it behaves any better.
I have a question for a biology question. Aren't eels fish?
I don't know.
I'm trying to spell
yes.
Bathioconger. I'm kind of distracted.
Okay,
Anyone have ideas about that?
I'm guessing BATHY. Some Euro. And then Conger, CONGER.
Yeah, it's just not looking as good as it did yesterday.
Yeah. No.
But
We'll go with that.
yes, I understand your thing about
what is
the
that right ahead
fish
of
and
us?
the eels.
Eels are
Zoom.
ray-finned fish.
Yes, they are.
So wouldn't a fish like eel just be an eel?
Zoom in there.
Coming in.
Another holothurian. I shouldn't say
Oh,
just another.
I thought that
Nice
that was
baby.
in the
Aw,
water
pretty
column.
one.
I did too for a second.
It's a pretty
Beautiful color.
I'm gonna open up the iris kind of just to get a look at it. It's gonna blow out the sand. Sorry for your eyes. Yeah. Very cool.
So it has something living on it.
Full zoom. Oh, a little too tight.
Looked a little shrimpy.
Coming in a little tighter here.
All right, I'm full wide on it. Get out of there.
Full wide.
Beautiful.
So I think more accurate would be a bony fish like
Yeah.
eel.
That's what I was thinking too.
I see. Yep, I see.
Do you?
Well,
Yeah, I guess I would be like, hey,
Yeah.
that whale kind of looks like a mammal.
A little bit. But fish are complicated, and I'll leave it at that.
My fisheries biology instructor is one of the internship coordinators at the academy, so if I start getting this stuff wrong, they're just going to yank me off the boat.
you think that person is listening? May well be.
Maybe.
May well be.
Well, they can definitely watch the dives on YouTube in a few days and check up on your...
Yeah, hi, Dr. Murakovich, if you're watching.
It's a good name.
It is. It's much like yours, very complicated. It's got like a RCV. So yeah, it's I don't know.
Mm-hmm.
Yeah.
Lots of letters.
Yeah. It's like you're playing Wheel of Fortune and couldn't afford any more vowels.
That's the joke that my parents have been saying about our last name forever. Or that someone tripped, fell, and spilled a bowl of alphabet soup. Sponge to the right.
Oh yeah, nice one.
It's got its own little peak there above the rest of the...
We're still below 2,500.
Zoom in there.
We gonna
Coming
want
in.
this one?
You want to sample it?
Yeah, it's different than the other one, so let's.
Okay, stop
What do you
ship.
think, Jamie?
Bridge now.
I think let's do it.
Okay.
We've
Request
only
hold
gotten
position.
one sponge sample this cruise aside from the one we took earlier this dive, so especially since we're interested in ones below 2,500, I think this is a good opportunity.
Okay, full wide. I could try and get that with the coral cutters.
Yeah, it's gonna be pretty floaty, and it's another glass sponge too, so it's gonna. If you think slurp might be better.
Yeah, we can slurp. Can you, Kylie, punch up the slurp camera and rotate the jars around to the flush jar?
Are you sure?
Looks like it's on flush.
Do we have the same mesh on all the slurps?
Yeah,
Okay.
yeah.
It actually looks like there's a different one on flush than there is on the next one over.
Flush has no mesh.
Yeah.
Oh, I see.
All
okay.
the other ones have the same mesh on this
Okay.
currently. Okay, I'll land out.
If we have time with the ship move, a quick zoom before we slurp would be awesome.
Roger.
Ship is currently holding position.
Okay. I guess I know it's holding, but I don't know how much swing we're worried about. But yeah, if we have time, that would be great.
Can you look down with the Argus camera? Just try and keep me in the view there. Keep an eye on all that, how close Argus is coming in.
I'm paying
And what sample jar are we on? Looks like
I don't think we've
We
slurped
haven't
anything
we
yet.
haven't slurped anything, so pick your favorite.
right.
Except for flush.
I just want to see it. Am I getting the numbers right? That looks like 6 and 7. That's 1. Okay. Number 1.
Sounds great.
Do you want to zoom before, you said?
Oh, right. Yeah, we need to zoom. Sorry. Go ahead with that.
That's all I got for down.
Okay.
All right, we're good. Thanks.
Okay, full wide.
Looks like there's somebody inside.
Full wide.
I was just going to say, I wonder who's inside there.
Kylie, you ready to do the suction pump?
I'd guess a shrimp, but we'll see.
Yeah.
Hook hydraulics, and it's gonna be T2, right?
Yeah.
Yeah.
Kylie, let's just do a slow ramp up on the.
Yeah.
The winch as
Take it.
well. Gotcha.
Take it up to 30 or 40%. Zoom in a bit there, video.
Okay, let me know when you're ready. You want it now?
Yep.
You've got 40.
Hold on. I'm too close to that. Okay. A little more.
A little more?
That might be okay. Hold on.
Okay, well, you're at 50.
There it goes.
All right, just let it all get in there. Okay, shut it off.
Off.
Nice.
There it is.
There you
And
go.
its friend.
Yeah,
And then
it's-
rotate it
shrimp.
back so that the flush jar is in the
Okay.
ready to go.
Full one?
Yeah.
Oops, wrong way.
Stayed pretty well intact too.
Yeah, surprisingly. And
Yeah.
little fish in there.
Okay.
Very
Shrimp.
nice.
Always shrimp.
You guys all set?
Good job. Yeah, ready to go.
Bridge nav?
Nav, can you drop a target?
Yes, I can. Request we continue tracking our line.
This is 037.
037.
We've got
You want
some.
your starboard rail cam back?
No, that's okay. Sure. Yeah.
We've got some viewers wondering if your skillful ability to use all of the tools on Hercules has garnered you any luck with the claw machines on the boardwalk.
Ooh.
Yeah. Sorry. Maybe I should have a go at them again.
I mean, I'm pretty sure those are rigged though.
They're
so.
pretty rigged, I think. Yeah.
Yeah, they turn the pressure down on its grabbing ability so it can't hold anything hard enough to pick it up.
Yeah, and then these slippery little fingers that are meant for things to fall out of. But you're right, it couldn't hurt.
The claw.
Give it a try, but don't stay too long at those machines. Don't spend all your allowance on it.
Much prefer a pair of long tongs.
Our favorite tool. And in terms of collections being retrieved from Hercules, at the end of these dives, when the vehicles are back on deck, that's when our science team leaps into action to take all of the samples out of the bio boxes, slurp containers, get them in the fridge, get them labeled and processed. And when the vehicles are retrieved, usually you can get a peek of that by watching the quad view, because we usually have the wet lab cam on, and you can see all of them, you know, imaging the samples, again, labeling them, processing them in whatever way each individual sample needs to be processed. And again, for our viewers watching on nautiluslive.org, thank you for tuning in. We are, yeah, it looks like a
Another
larger sponge.
Zoom in there.
We are south of Palmyra Atoll. Cool.
Oh, that's a neat one.
It almost looks like one of those
Come in
chubby
tighter.
slime stars at the top,
Sure.
even though I know it's a sponge.
Are those little tiny sponges growing in the sediment in the background? What are those?
Huh.
I think that's what I was seeing off of those rocks a while back.
Yeah, I've been seeing these tiny little, just white, almost cones on top of some of these rocks.
Don't think they're sponges. I think they're harder than a sponge. But I'm not sure what exactly they are.
Okay, come wide.
Nav, when you drop high pack targets, does it record depth as well?
That is a solid question. Let me go see if I have one on the last one.
Oh, sorry, I was drinking tea. Forgot about it.
Distance bearing.
Oh, there's some coral on the right.
No, we do not have a depth.
Okay, no worries, thanks. Where's the coral?
Yeah,
On the
to
bottom
the
right.
Yeah.
Oh, wow.
Out of screen.
Totally missed that.
Yeah, Kevin's got an eagle eye back here.
Yeah. Want to adjust to this kind of little blowy screen that we got.
Jamie, if you want, when we get samples, I can
come in there, video.
Coming in.
read you off depth for the notes.
Yeah, I normally don't write down because it is
Whoa.
auto-recorded. I was just going to add it in since we were particularly interested at the particular depth for sponges, but I can pull it from the notes later.
Roger.
So, no worries.
What's on this one, Jamie?
It's very interesting.
Come in tighter.
I
This
think they're barnacles.
wow.
Is that full zoom?
That's full zoom there.
Okay.
Is that something that you would want to sample?
I don't know. I know we haven't gotten any corals on this dive actually, so yeah, I imagine Steve would like a snip of the coral. We wouldn't need the whole organism.
Maybe snip the piece with. I mean, 1, 2, 3.
Looks
4.
like
Yeah.
Too many on that branch.
Well, if we did the
can you snip
between the two on the
clusters?
stalk and the few higher up, I think that would be an
stop
okay
shifting.
number.
Bridge nav.
If it's possible to get the coral
Crest hold
clutters
position.
in between
In
those
between
two.
those two.
Yeah,
Mhm.
between the two adjacent to each other on the stalk and then the large cluster out there. If it's possible to snip there, that would be ideal.
Yep. Okay, full wide video.
And maybe a snip and slurp. If you think that would go up the tube okay.
Yeah, it should. Yep.
The whole branch, or just slurp the barnacles off?
The branch too.
Oh, okay.
Yeah.
Come up on Argus a little bit, Kylie, as he slides in there.
Thank you.